The water is black, not the darkness of night, but the absence of light itself, a cold, crushing void where the sun hasn't touched in months. Beneath the Arctic ice, the ocean hovers just above freezing, a place so hostile that ordinary blood would crystallize in seconds. Yet here, in this frozen cathedral of silence, life moves. Slowly, deliberately, fish glide through water that should kill them, their bodies defying the fundamental laws of chemistry. Their blood flows clear, not red. Their hearts beat once every 10 seconds. Some have no hemoglobin at all. Others produce antifreeze in their veins. The closer you travel to the poles, the stranger these creatures become. Not through randomness, but through millions of years of evolutionary negotiation, with an environment that refuses compromise. Today, we're diving into the biological extremes of polar fish, where survival demands anatomical impossibilities, and where the cold has sculpted life into forms that challenge everything we thought we knew about vertebrate biology. The story begins with a simple, brutal fact: ice crystals are lethal. When water temperature drops below 0 degrees Celsius in seawater, around -1.9 degrees, ice doesn't just form on the surface. It seeds itself from within, nucleating around any particle, any imperfection. For a fish, this means death from the inside out. Ice propagates through tissues, rupturing cell membranes, shredding organs. Ordinary fish, the kind you'd find in temperate oceans, carry blood and cellular fluid that freezes readily. They cannot survive in polar water for more than minutes. Yet the Antarctic toothfish glides through the Weddell Sea at depths where the temperature never rises above -2 degrees. The Arctic cod swims beneath pack ice for its entire life, never knowing warmth. These fish don't avoid the cold. They have become it. The solution lies in antifreeze glycoproteins, molecules so strange, so specific, that they exist almost nowhere else in nature. These proteins bind to the surface of forming ice crystals, preventing them from growing. They don't stop ice from appearing, they freeze the process of freezing itself, locking the crystals at a size too small to damage tissue. The Antarctic nototheniod fish produce these proteins in their liver, circulating them constantly through their bloodstream, through their eyes, through the fluid surrounding their brain. The proteins are encoded in genes that evolved between 5 and 15 million years ago, when Antarctica's climate shifted from temperate to frozen, and the ancestors of these fish faced a stark evolutionary choice: adapt or vanish. Most vanished. The notothenioids adapted, and today they represent more than 90% of fish biomass in Antarctic waters. They didn't just survive the cold, they inherited it. But antifreeze proteins are only the beginning. As you move further north or further south in the Antarctic, the cold doesn't just threaten with ice, it slows everything. Metabolic reactions depend on molecular collisions, and in freezing water, molecules move sluggishly. Enzymes that work efficiently at 10 degrees Celsius become nearly inert at 0. Muscle contractions slow. Nerve signals lag. Oxygen becomes harder to extract from the water, even though cold water holds more dissolved oxygen than warm. The fish are caught in a biochemical paradox. Their environment is rich in resources, but their bodies are too cold to use them efficiently. Evolution's answer has been to strip away what isn't essential, and to redesign what remains. The blackfin icefish, Cainocephalus aceratus, swimming in the Southern Ocean, has no red blood cells, none. Its blood is translucent, a pale yellowish fluid that moves through its body carrying oxygen dissolved directly in the plasma. Hemoglobin, the iron-rich protein that gives blood its color and its oxygen-carrying capacity, is absent. The genes that code for it have been deleted, rendered non-functional by mutation. For any other vertebrate, this would be fatal. But the icefish survives because the Antarctic Ocean is oxygen-rich, because its metabolism is slow, and because it has compensated in other ways. Its heart is 3 times larger than expected for its body size. Its blood volume is 4 times greater. Its capillaries are wider, reducing resistance. It moves slowly, conserving energy, existing in a state of perpetual metabolic efficiency. The loss of hemoglobin may have even been advantageous. Hemoglobin increases blood viscosity, and in the extreme cold, thick blood becomes dangerously sluggish. The icefish solved this by eliminating the problem entirely. Then there is the question of buoyancy. Most fish maintain neutral buoyancy, using a swim bladder, a gas-filled sac that adjusts to changing depth. But gas is compressible and temperature sensitive, and in the deep, frigid polar seas, swim bladders become unreliable. Many polar fish have lost them. The Antarctic toothfish, which can weigh up to 100 kilograms and live at depths exceeding 2,000 meters, has no swim bladder at all. Instead, it stores lipids, fats in its tissues. Fats are less dense than water and provide lift without the mechanical complications of a gas chamber. The fish becomes, in essence, a living oil reservoir, its flesh saturated with wax esters and triglycerides. This makes it slow, cumbersome, but it also makes it stable. It drifts through the abyssal cold like a zeppelin, conserving energy, waiting for prey. The further north you go into the Arctic, the stranger the adaptations become. Not because the Arctic is colder than the Antarctic, but because its environment is more variable. Antarctic fish live in stable, predictable cold. The water temperature changes little throughout the year. Arctic fish, by contrast, face seasonal extremes. In summer, surface waters warm slightly. In winter, they freeze solid. The fish must cope with both the cold and the instability. The Arctic cod, Borreogarda sida, is perhaps the most extreme example. It lives its entire life under sea ice, feeding on ice-associated plankton, spawning in winter beneath the pack. It can tolerate temperatures as low as -2.5 degrees Celsius. It produces antifreeze glycoproteins, but it also adjusts its enzyme function seasonally, expressing different protein variants depending on temperature. Its metabolism is flexible, shifting between states, optimizing for conditions that change slowly but relentlessly. And yet, for all their biochemical wizardry, these fish are fragile. They are specialists, exquisitely tuned to a narrow environmental band. The Antarctic notothemioids cannot survive water warmer than 6 degrees Celsius. Their enzymes denature. Their antifreeze proteins become toxic at higher temperatures, accumulating in tissues and causing cellular damage. The icefish, with their enlarged hearts and dilute blood, cannot generate the metabolic power needed for fast swimming. They are ambush predators, lying in wait, conserving every calorie. They are evolutionary masterpieces, but they are also prisoners of their own adaptations. There is something unsettling about this. We tend to think of evolution as a process that increases complexity, that builds towards something greater. But the polar fish remind us that evolution is also about loss. The icefish lost their hemoglobin. The toothfish lost their swim bladder. The notothenioids lost the ability to survive in warmth. They traded versatility for specialization, and in the frozen polar seas, that trade has kept them alive for millions of years. But it has also made them vulnerable. As the planet warms, as the Arctic ice retreats and the Southern Ocean begins to heat, these fish have nowhere to go. They cannot move to cooler water. There is no cooler water. They cannot adapt quickly. Evolution is measured in millennia, and the climate is changing in decades. They are relics of an ice age that is ending. What makes the polar fish so compelling is not just their strangeness, but what they reveal about the limits of life. Biology is chemistry, and chemistry is constrained by temperature. The proteins that fold correctly at 20 degrees Celsius unfold at 40. The enzymes that catalyze reactions in tropical fish seize up in polar water. Life exists within a narrow thermal envelope, and the polar fish live at the edge of that envelope. They show us how far evolution can push the boundaries, how inventive natural selection can be when the environment demands it. But they also show us the cost. The icefish's heart is enormous because it has to be. The toothfish is slow because speed is metabolically impossible. The Arctic cod cannot leave the ice because the ice is the only place where its biology makes sense. And there is mystery here still. We don't fully understand why antifreeze proteins don't harm the fish at higher temperatures, or how the icefish compensates for the loss of myoglobin in its muscles, which should render its tissues incapable of storing oxygen for burst activity. We don't know how some Arctic fish can survive being frozen solid, actually frozen, their tissues filled with ice, and then thaw and resume life when conditions improve. There are species in the high Arctic, small sculpins and snailfishes, that have been found encased in ice and revived in laboratories. The mechanisms behind this are only beginning to be understood. It involves dehydration, cryoprotectants, and a tolerance for cellular damage that defies conventional understanding. The polar fish are not metaphors. They are not symbols. They are real animals, shaped by real pressures, living in real time. They exist because the cold demanded it, and they persist because they have no other choice. The weirdness increases with latitude, because the cold increases, because the challenges multiply, because evolution is relentless and unforgiving. These fish are the result of millions of years of survival at the edge, where the margin for error is measured in fractions of a degree. They are not trying to be strange. They are trying to stay alive. And as we watch the ice melt, as we measure the warming of polar seas, we are left with a question that science cannot yet answer. What happens to a creature that has spent 5 million years adapting to the cold when the cold begins to disappear?